Good evening, everyone. Can you all hear me in the back? All right. My name is Julie Mickleberry, and I see a lot of familiar faces here tonight. I'm the Associate Director for Alumni Affairs with UC Santa Barbara, and I am so excited to welcome you to the third lecture in our Aging and Longevity series, Cellular Stress. Before I hand it over to Dr. Rothman to introduce the Center for Aging and Longevity Studies and tonight's speaker, I'd like to take a moment to express my gratitude to the CALS team, to my Alumni Affairs colleagues, and specifically to Diana Thomas with CALS and Dr. Nicola Abada with CALS, to Direct Relief for donating their wonderful space, to the Santa Barbara Independence, for helping get the word out about this lecture series. And we're so thrilled tonight to be able to bring the cutting edge research that many of my colleagues see happening every day on campus to the community. I also want to mention, I'm going to take a little liberty, our next lecture next month on April 24th actually lands during our All Gaucho reunion weekend. So I'm going to ask again tonight, how many of you in the audience are alumni? Fantastic. Thank you so much for coming out tonight. We hope to see you not only at Dr. Gervin's lecture on April 24th, but at some of our other events happening over the weekend. We have a downtown experience as well as a signature event called Gaucho Fest on campus. And it's a great opportunity to reconnect with some of your peers and to maybe make some new friends. So we hope to see you there. Now it's my great pleasure to introduce Dr. Joel Rothman, Director and Founder of the Center for Aging and Longevity Studies, and a distinguished professor of molecular, cellular, and developmental biology. Please join me at the podium.
Thank you, Julie. Thank you, Julie, and thanks for everything you've done to organize this. This has really been a wonderful series of lectures, and I think you'll enjoy tonight's. So many of you have probably already heard me talk about the Center for Aging and Longevity Studies, which was founded about 3.5 years ago at UCSB, a major science and technology center that's devoted to improving human lives through extending the period of healthy lifespan, our prime goal, preventing or forestalling age-related diseases, and guiding the impact of this longevity technology on society. We now encompass 43 faculty, so that's about 4 more than we had in the last lecture. Over 350 affiliated researchers spanning biomedicine, engineering, computer science, chemistry, physics, psychology, economics, actuarial science, social sciences, and the humanities. So we are one of the most interdisciplinary centers for aging and longevity studies anywhere. Our research is advanced by really cutting-edge, state-of-the-art facilities that help catalyze the discovery of preventive interventions in aging. Oh, sorry. Okay. Is that better? Yeah, normally I have a mic clip. I'm sorry if you missed all that. I won't start over again, though. We're also developing a clearinghouse of reliable information that's based on scientifically rigorous foundations. We'll be announcing that later in the year. So if you look into aging or longevity science by Googling, you will find an enormous amount of stuff that it does not have strong scientific foundations, if you'll forgive me, but it is true. Perhaps 90% of it. So we're going to counter that with a rigorous clearinghouse that is directed by practicing scientists who will use the scientific literature and scientific foundations to advise you on what might be and what might not be appropriate ways to think about what you might do to extend your own longevity, healthy longevity. Okay, and now it's my privilege to introduce tonight's speaker, a close colleague of mine, Denise Montell. Professor Denise Montell received her bachelor's degree in cell biology from UC San Diego. Unfortunately, not an alum of UCSB, but you know, if you spell it wrong, she's here. She has a PhD in neuroscience from Stanford and performed postdoctoral studies at the Carnegie Institute of Washington. For 20 years, she was a professor at Johns Hopkins University Medical School in the Department of Biological Chemistry, and she served there as the founding director of the Center for Cell Dynamics, and some of her current work still, I think, ties into that earlier work that she did at Hopkins. And while I was chair of molecular cellular developmental biology at UCSB, I was very fortunate to be able to recruit Dr. Montell to UCSB to the Duggan Professorship of Molecular and Cellular and Developmental Biology. And I must say that bringing Denise here to UCSB is one of my proudest accomplishments as chair of the department. She's done an enormous amount to advance biomedical science at UCSB, including in CALS, I might say. Dr. Montell's research focuses on revealing how cells collectively collaborate to make an animal function properly. And this includes her vital contributions to understanding how groups of cells move through tissues, actively can move through tissues, how cells gobble up undesirable cells, which is very important, for example, in cancer therapy. She may say something about that tonight. She's also investigated how cells can be brought back from the brink of death and revitalized. And this evening, we'll also discuss how animals become resilient to stress. Those last 2 points particularly are of prime relevance to the processes of biological aging that cause the process of biological aging in animals. Denise's research has pointed to novel approaches in the treatment of diseases, including elimination of tumor cells and promotion of health span, starting with model organisms in the laboratory. I can think of almost no one who's done more to transition from the laboratory models to true interventions in human biology, human biomedicine than Denise Montell. Her research accomplishments, as I think you can gather, place her among the ranks of the top biomedical scientists in the world. And in recognition of her contributions, she was elected a Fellow of the American Society for Cell Biology and the American Association for the Advancement of Science. She served on advisory councils for major institutes, for 2 NIH institutes, the American Cancer Society and the Howard Hughes Medical Institute. She is a 2-time winner of the most coveted, I think, award from the National Institutes of Health, the Pioneer Award, the NIH Director's Pioneer Award. One of the most competitive grants to get nationally from the NIH. She got 2 of those, the latest just funded in the last year. She has been the recipient of the 2024 E.B. Wilson Award of the American Society for Cell Biology. And finally, in 2021, she was elected to the U.S. National Academy of Sciences, among the highest recognition that a scientist can receive. So it's a great honor and privilege to introduce to you Denise Montell. Please welcome Denise to the podium.
Okay, well, thank you so much. Sounds like you can hear me in outer space. It's a pleasure to be here tonight to tell you about some of our work that we're doing. And I want to start by asking you to imagine with me a near future when cellular immunotherapies would be inexpensive and easy to deliver. And I want you to imagine a tiny particle that you could inject that would program your own immune cells to seek out and destroy tumor cells anywhere they would be in your body. And not only tumor cells, but kind of anything, any cell or substance that was undesirable for your body. And so this is the technology that my lab is really working on and developing based on our earlier work that started more than 30 years ago, working in fruit flies. So in my lab, we're really fascinated by cells. And we're interested in how cells behave normally when they're building and maintaining and repairing tissues, and then how cells misbehave in diseases like cancer. And so our goal is to understand cell behavior so well that we can manipulate it and harness it to treat disease. So to back up a moment and remind you of things I'm sure you already know, I just want to remind you that our bodies are made up of cells. In fact, human bodies are made up of trillions of cells, maybe between 30 and 40, depending on how big you are. This microphone keeps falling off my ear, so let me see if I can get that fixed. Okay, and each cell has a nucleus, and inside that nucleus are the chromosomes. And the chromosomes contain your unique DNA. And this is where our genes reside. And I think people are probably pretty familiar at this point with the fact that the DNA encode, is a code. It's a chemical code. And this code is read out every day in every cell by converting the DNA sequence into another molecule called mRNA. And this mRNA is then another code that's read out into a sequence of amino acids to make up a protein. So we have something like 23,000 genes in our genomes that encode more than 23,000 proteins, since each gene can encode a couple of different forms of a protein. And this is what, when this sequence of chemicals that are amino acids fold up into its final structure in your cell, it's going to look something like this. Now, these are actual protein molecules inside of a cell, a real cell. And these are protein molecules that they're not normally, they're not color-coded inside your body, but they're color-coded here. And you can see that, the cell is chock-a-block with these protein molecules, and they do some amazing things. They assemble into sort of highways and, train tracks and all kinds of fancy little particles. And they're, each one has a unique shape, and it's carrying out its unique function within the cell. Now, like I said, we're really interested in cells, and we're interested in how they behave. And a lot of our studies really start with this model shown here, this experimental model that you can see here. And what this is an ovary from a fruit fly. So inside of a fly, it looks something like this. But if you spread it out on a microscope slide and look at it with fluorescence, it looks like this. And this is an amazing model. We love this model because there are incredibly diverse biological questions that you can address and cell behaviors that you could see. And then there's this beautiful and simple anatomy that makes it accessible to us to see. There are really powerful genetic tools that we can apply in this organism that you really can't apply in other organisms. And then there's a long and rich history of success in using this organism to make fundamental discoveries. And as a little aside, here are the 9 individuals who've won Nobel Prizes for their work in Drosophila, in the fruit fly Drosophila, figuring out, that genes are on chromosomes, learning how genes are deployed to carry out embryonic development, like you yourself built yourself from a single cell without any instructions other than the ones in your DNA. And that was sorted out using the fruit fly. There's been an award for understanding the molecular mechanisms of immunity and for insights into animal behavior. Well, like I said, in my lab, what we're interested in is cellular behavior. And so if we zoom in on one of these tiny little developing eggs in this ovary, we can see something like this. And I think you can appreciate that this little group of cells right here, so this is a group of about 6 cells, and this is a cell, and this is a cell, and this is the growing egg cell, that these cells are traveling in a process that very much resembles the way that tumor cells spread through the body in the process of metastasis. And so we've been studying this. I got interested in this behavior in 1988. Okay, so I've been working on this for a long time, and you might wonder how in the world anybody would study 6 cells in a fly ovary for that long. And yet I hope by the end of the evening you'll understand why. But mainly we were just super curious about how this worked because it looks fascinating. And at the time, we didn't know a single thing about it, literally not a single thing, not a single gene that controlled it, and we couldn't do the kind of movie that you're seeing here. And so I set out to identify the genes and the proteins that are required for this process. So what makes these cells move and how do they do it? And so we decided, looking back at this kind of genetic cell here, we decided to focus on this protein right here. Okay, let's see what this one does. And you might wonder, why would you pick that little nondescript cyan dot in the middle of that thing to look at? And the reason is that, just moving.
The member sling
Um,
example is a cell which.
So the reason is because the very month that I started my first faculty position at Johns Hopkins, this paper came out, and it's a classic in cell biology. And Anne Ridley and Alan Hall showed that if you add an active form of this protein whose name is RAC to a resting cell, that it does this. It starts doing this ruffling and dynamics and so on. And we didn't really know much about how cells move, but this seemed like maybe it had something to do with it. So that's why we picked this protein called RAC. And in fact, RAC is really, really ancient. It's as ancient as complex cells. We can find it in the tree of life going back to 1 billion years ago when the first complex cells evolved. So, so we wanted to ask the question, is RAC required for cells to move? And the way to do that is to block the activity of RAC in a cell and ask what happens. And this is what my very first graduate student, Anne-Marie Murphy, did. And so you can see here these little cells that I was telling you that are migrating along. And you can see here what happens if you block the activity of RAC. They cannot move. So that was exciting, and it was the first demonstration of that. And now we know that RAC is required almost universally for cells to move, from a single-celled amoeba to a human cancer cell to a human immune cell. RAC is really fundamental to this process of cell movement. But we had a real puzzle early on when we were studying this protein. And so here I'm showing you these cells before they start migrating, and they're sitting there expressing this fluorescent protein, but otherwise they're normal. But if we express a hyperactivated form of RAC in these cells, you get what happens here. And I think anybody can see that this thing is alive and well, and this thing is dead, okay? And so that was puzzling to us because we were putting the same form of RAC in these cells that Anne Ridley and Alan Hall had put into their fibroblast, their skin cell, and that cell had danced and ruffled and everything in response, and actually drinks fluid in response to this protein. But here, this entire structure, this entire tissue was dying as a result of our adding active RAC to these cells. And why and how that was happening was a mystery to us for 25 years. Okay, we called it the cold case in the lab. We just didn't understand what was going on. And we weren't always thinking about it because it was kind of cold, you know? But then eventually we figured out that the reason this was happening is because when we express hyperactivated RAC in these green cells here, whereas normally our migrating cells, you know, they're extending outwards in the direction they're migrating, and they're kind of carrying these pink cells along with them. But when our cells express hyperactive RAC, they turn on them and eat them alive. Okay, and so we're quite sure this is why the tissue is dying, is because these cells are eating up all their neighbors. And that's because if we block their ability to engulf their neighboring cells, then the egg chamber, this tissue lives. These cells are still expressing hyperactive beta RAC, but now this whole structure is alive because we've blocked their ability to eat. So border cells expressing hyperactive RAC kill this developing egg by eating it alive. Okay, now that sounds pretty gruesome, and you might think that's some kind of weird fly business going on, but actually, in your body, you turn over 2 million red blood cells every second of your life by this very process. Okay, so in your liver, there are cells that eat other cells, and they can recognize the spent red blood cells, the used up ones, and they remove them by eating them alive. So this is an extremely common process. Your white blood cells will also do this when you resolve an inflammatory response after an infection. So you get an infection, you make all these blood cells to fight that infection, and then when the infection wanes, you have to get rid of all those extra cells that you made, and this process gets used again. So we have cells in our bodies that vacuum up debris, and they're called phagocytes. This is like from Greek roots, phago means eat and cyte means cell. So these are phagocytes, and they're cells that vacuum things up, and they vacuum up things like viruses, as shown here. They vacuum up bacteria, virally infected cells or bacterial infected cells, dead cells. Some of our cells die, or whole red blood cells. And this process depends on RAC. RAC is one of about 150 proteins that participate in this cell eating process. And so we had found that in the fruit fly, adding hyperactive RAC to a cell was sufficient to cause that cell to eat another cell alive. And because RAC is so ancient in evolution and our cells have RAC too, we decided to test whether that was also true in mammalian cells. So this is the work of Mel Rodriguez. She's a PhD student in the lab. In this movie that you just watched, the macrophage, and a macrophage is a special type of eating cell. It means big eater, macro, big, phage, eat. So it's a big eater, it's a big cell, and it eats other cells. And these little cells in this example are T cells, which are a white blood cell, and they're labeled in magenta. And then they're also labeled with a dye that's going to turn yellow if that cell gets eaten up. And so these are normal cells, okay? And in these normal cells, there's not a lot of eating going on. Now, this is what happens when the cells have a hyperactive form of RAC. And I think that you can see that there's a lot more eating of these living T cells going on. So, okay, so that was really cool because we found something in the fruit fly that turned out to also be true in mammalian cells, including human cells. So mouse cells, which you just saw on that slide, and also human cells. So then we wanted to know, can we harness this activity, this improved eating of another cell, to treat cancer? So I'm sure many of you have heard about CAR T therapy, but just in case anybody doesn't know, the strategy with CAR T therapy is to extract a certain type of white blood cell called a T cell from a cancer patient and engineer that cell in the laboratory to express a molecule on its surface that will target it to cancer cells. So it looks something like this is your CAR T cell, and this is the thing you've engineered it to make, and it's on its cell surface, and it's recognizing the tumor cell through this specific interaction. And so then one engineers the cells that way in the lab, expands them, and reinfuses them into the patient. And this has been really quite effective against a variety of types of cancer, including blood cancers. But there's still some limitations to CAR T therapy. And one of the therapies limitations is really with solid tumors. So tumors like breast cancer, ovarian cancer, lung cancer, colon cancer, these are common cancers, but those tumors, the reason they're called solid is that they make this hard shell around them. And the CAR T cells have a hard time getting through that shell. But there are other cell types, and macrophages being one of them, that are really good at getting inside of the tumors. And there's some other limitations that are limiting how much we can use CAR T therapy. There can be, not every single tumor cell necessarily has that target that you're going after. CAR T cells can become exhausted. That's actually a cellular state. And there can be some side effects. And so people are working very hard to overcome these limitations. And one approach that's being taken is a newer cousin of CAR T, which is called CAR macrophage. So that's where we take, the idea is very similar. Now we're going to take our macrophage, our big eater, and we're going to engineer it in the lab to express a molecule that's going to allow it to recognize a tumor. And then the hope is that what's going to happen is that the macrophage is going to eat that tumor, and then also be able to send signals to activate the rest of the immune system to recognize it. And the hope is that this can generate potentially a broader and more durable response. So This idea is exciting, and first in human trials are underway, and it's very clear that this therapy is safe. There are no, so far, dose-limiting toxicities, but it's also clear that it could be more effective. But I first heard about this from an assistant professor in our department of MCDB at UCSB. She came to give a job talk. And she showed, these are her movies. This is Megan Morrissey. And here we have our macrophage in green. And in this case, a B-cell lymphoma cancer cell in magenta. And we haven't engineered this macrophage. So it's, touching the lymphoma cell, but it's not doing much. Now, Megan adds this CAR, this molecule, to allow it to recognize the tumor cell. And now we get the macrophage eating tumor cell. But more frequently, what happens is that the macrophage kind of nibbles on the cell and doesn't actually totally eat it and kill it. And it's sort of taking little bites. And so it's very clear that enhancing the ability of a CAR macrophage to engulf and kill a living target cancer cell would be really helpful. And so when Megan came and gave her presentation, of course, immediately what leaped to mind to me was, why don't we add an active version of RAC to this CAR macrophage? Because we know that RAC enhances the ability of cells to eat other cells. And our hope is the CAR is going to tell it what to eat, the tumor, and the RAC is going to enhance that eating. And this is what we saw when we added active RAC to the macrophage. So now it had the ability to gobble up a whole bunch of tumor cells. So in fact, it looked like with RAC alone, we didn't get this effect. We had to have the CAR to tell it what to eat, and then we add the RAC and it enhances the ability to eat. So we call this RAC enhanced CAR therapy or race CAR therapy. Okay, so we're very excited about this, and it's what the latest Pioneer Award is for. And I want to show you that, because we think the receptor tells it what to eat, and the RAC enhances that eating, it could be quite generalizable to different types of cancer, and also not necessarily only cancer, different types of targets that you might want to eliminate from the body. So let's test that idea. And so Elise Dezek, a student in the lab, was interested in glioblastoma. This is a pretty bad cancer, and there's a great medical need for treatments. So in this movie, these are normal macrophages, and these are glioblastoma cells in magenta. And there's no CAR and there's no RAC being added to the cells. And what happens over the course of a couple of days is that the cells ignore each other, and the glioblastoma cells migrate around and they divide. This is what happens when we co-culture race CAR macrophages together with the glioblastoma. Now, these race CAR macrophages are expressing receptor that makes them target glioblastoma specifically. And you can see by the end of the movie that there's hardly any glioblastoma cells left in the culture. So, like I said, we think we could generalize this sort of form of personalized medicine. CAR T, the nature of what CAR T does, allows it to attack cancer cells, but not necessarily all of these other things that we think CAR macrophages could eat up. So they, fungal infections can be really hard to treat, but we think race CAR macrophages could, in fact, attack fungi, amyloid. We know we don't like to have amyloid building up in our brains. We think that race CAR macrophages could be programmed to eat amyloid, different diverse types of tumors. And we've seen in the lab that we can see effectiveness against lymphoma and glioblastoma, also triple negative breast cancer cells. And we're moving those studies through collaboration with folks at the Huntsman Cancer Center at the University of Utah and at City of Hope. We're working to test these ideas in mouse models of these diseases. But we also think we could target things like fibrosis and antibiotic-resistant bacteria and other types of targets. So In the last couple of minutes or last minute or two, I just want to mention a really important limitation of all the cellular therapies right now. And a really big limitation is the complexity and cost of these therapies. So that procedure that I described to you of taking cells out, engineering them in the lab, expanding them and reinfusing them into the patient, this can cost like $1 million per patient. This is not scalable. It's not accessible. And so this is a huge problem for all of the cellular therapies. So we have an idea of how we might overcome this most serious limitation of these therapies. And so the idea is sort of like this. We had the incredible success of the COVID vaccines, the mRNA vaccines. Whatever you're hearing else to be in any other direction, we had really incredible success, and they were scalable to the entire world. Okay, and the idea there was to take an mRNA that encoded the spike protein from coronavirus, encapsulate it in lipid nanoparticles, and deliver that to people. That's what you got in your COVID shot. Our idea is to take our RAC mRNA and our CAR mRNA, encapsulate them in these lipid nanoparticles, and then we would have a different CAR for each disease that we were targeting: a breast cancer CAR, a glioblastoma CAR, an antibiotic-resistant bacteria CAR, a CAR to target each disease. This would now be an off-the-shelf therapy rather than an individualized therapy for each individual patient. And this would reduce the cost. And so then this would get injected, infused into the body, and now the macrophages are naturally big eaters. They're the cells that are going to go eat those lipid nanoparticles above and beyond any other cell type. They're the ones who are patrolling the body, looking for things to eat, and they're going to eat it, express the RAC and the CAR, and then keep patrolling the body and find those targets that we want to eliminate. So call that infusible or iris CAR. Okay, I just want to stop there and thank, I've tried to acknowledge some of the people in the lab. Abhinav Mishra is a postdoc in the lab who's about to set off on his own adventure. All of these people make an incredible team. Megan Morrissey, I couldn't have done any of this work without collaborating with Megan. And I want to acknowledge our very important funding from the National Institutes of Health, including the National Institute on Aging and the National Cancer Institute. And I will stop there and take your questions.
Radiation treatment and chemotherapy a thing of the past? Could it make radiation and chemotherapy a thing of the past? Well, that would be an optimistic view of what it could accomplish. One thing that we have noticed is that, I mean, as you know, most cancers get treated by some combination of things, combination of chemotherapies, combination of radiation chemotherapy, surgery, radiation chemotherapy, because it's very hard for one therapy to really wipe out every cell. And one of the things that we've seen in our experiments with triple-negative breast cancer cells is that we can combine the RACE CAR with a drug that's currently in clinical practice. It's called olaparib, and it specifically targets the BRCA mutant cells. And what happens is olaparib is very effective against triple-negative breast cancer and ovarian cancer with these BRCA mutations, but invariably people develop resistance to that single agent. So what you really want to do is have a combination of things where you can make sure that, that'll reduce the chance that resistance will develop against that single agent. So we think that these RACE CAR approaches will, they may be effective as single therapies, and then, but we have evidence that they'll be effective in combination with other therapies that might prevent that relapse.
When you get these macrophages, really going crazy, eating up bad things, like some viruses and infectious things, doesn't that set off a whole inflammatory response in your body?
Mm-hmm. Yeah, interesting. So The cells themselves are in a hyperactivated state. And there are two well-characterized states of macrophages, the sort of wound healing, like healing promoting macrophages. So if you get a wound and there's some dead cells there and macrophages need to come in and clean that up or get rid of the immune cells once the wound has healed, that's a sort of pro-healing state. And then there's this inflamed state that they get into if there's a tumor or an infection or something that they need to fight more aggressively. And what we see is that our enhanced macrophages are neither in the one nor in the other. So they're not really hyper-inflammatory in their nature. They're hyperphagocytic. And so we can actually also program them to do the wound healing kind of behavior. So by giving them the right receptor, we can send them off in the direction of promoting wound healing. So we're testing the idea that they might actually be helpful in promoting the healing of chronic wounds, some of which fail to heal because there's excess dead white blood cells in the wound. And so I don't think we're not terribly concerned that they're just going to set off, a hyperinflammatory response. We have some evidence. There are actually human patients who carry activating mutations, like germline activating mutations in one of the RAC genes. And rather than having a hyperinflamed immune system, they actually have an immunodeficiency. And we can see, but by and large, they develop normally and their tissues are fine. But in that case, their macrophages eat their T cells, and this causes an immune deficiency in the patients. And what we figured out was that their macrophages were hyperactivated for eating, but their T cells, because of the RAC mutation in the T cells, was making them super sensitive to being eaten. So the main phenotype in those people is not hyperinflammation, but immunodeficiency. And we think that's mainly because of the way hyperactive RAC is acting in the T cells. Long story short, we're not too worried about these hyperactive macrophages bad in some way.
Have you been testing with humans?
No, our next step is to test in animal models of cancer, and so we're collaborating with folks to do that. So we're moving into animal models. So far, we've done this in cells.
When macrophages eat, when there's nothing left to eat, what becomes of them, and the other could this somehow be manipulated to be a vaccine?
Okay, so what happens to the macrophages after they eat? Macrophages live in your body about 9 days. So they're going to die and be removed by other macrophages eventually. So we think the therapeutic macrophages, will deliver them. They'll do their thing for maybe 9 days. Well, we need to give them more than once. We don't really know, but they're not cells that themselves grow and proliferate and so on. So we're not worried about the therapeutic cell like itself becoming a cancer, which is also a potential very rare but side effect of CAR-T. So, but what happens to the cells is that eventually after a week and a half or so, they die. Can it be used as a vaccine? Okay, so this is a little deeper into immunology than maybe everybody wants to get, but in principle, yes. So when a macrophage eats another cell, like a tumor cell, let's say it's eating the tumor cell, it digests it up, gobbles it up, and uses enzymes to digest that material. And then it puts all of that tumor stuff on the outside of itself. It displays it to the rest of the immune system. And that's called antigen presentation. So that's a way of, and most of the proteins that are in a tumor cell, they aren't from your own body. They are yourself, right? So most of the things that the macrophage is putting out there on the surface are just self, and that's not going to do anything. But tumors have a few strange proteins that are particular to tumors. And when those get put on the surface of the cell, it activates the rest of the immune system to recognize that tumor as foreign to be eliminated. In that sense, it's possible for the race car macrophages to stimulate more of a type of a vaccine against relapse.
Are there any types of fasting that can help with this process?
Fasting, like nutrient fasting.
Fasting would be really,
I am unaware of anyone who does that. Oh, sorry, the question was, is there any chance that like fasting, depriving yourself of nutrients would impact this, the effect of these cells one way or the other? And I'm not aware of anyone studying.
you know, in my years of teaching physiology, I always told my students I was going to come back in my next life as a macrophage. And now, after listening to all of this tonight, I realize I'm now going to have choices of what I'm going to be paired with or what kind of macrophage I'm going to turn next.
And would you want to be a race car?
What level of concern for continuing federal funding?
My level of concern is very high. Okay, so obviously, like what's been going on in Washington is really scary for everybody working in science. And so we got a very large award to work on this project because we're really excited about it and it looks really promising. And it's kind of an out of the box idea, which is why I got this Pioneer Award. I'm really concerned. So we're in year one of it, and we got, $700,000, and I hired some people to work on the project, thinking that we'd have that money each year for 5 years. And now I'm really worried that the second increment in the fall won't come through. And then what do I do with the PhD students who are supposed to be working on this project? And then it all stalls, and the NIH has invested many millions over the 30 years of my career in this project to get it to this point when we're almost there. And to pull the funding out at this point would be anything but efficient.
My question is, would you speculate on how these researchers that you discussed tonight provide a takeaway for those concerned about longevity?
Yeah, sure. And I should have mentioned that earlier. Good point. So how does this relate to Cal's mission? Well, the question is, this is a series on longevity and aging. And so what has this got to do with longevity and aging? He was more polite than that, but that was basically the question. And I want to point out here a couple of things. Actually, I have a slide on the hallmarks of aging, but as you know, cancer and neurodegenerative diseases, these are diseases of aging. And, I think race cars could, down the road be used preventatively. So for example, there are antibodies against amyloid, and you could program a race car macrophage to eat up the amyloid before it was doing really awful things. In addition, not shown on this slide, but another type of cell that is doing you no good as you age, many of us, and good as we age, are senescent cells. And so this is a type of cell that, okay. Oh, it is on my list. Thank you. It's on my list. It's not in my diagram. It's right here. So as we age, we accumulate senescent cells, and these are cells that have stopped dividing, and they're not particularly functional, but they're taking up space, and they're secreting inflammatory factors, and they're causing us harm. And so there's evidence in animal models that if you remove senescent cells, that you can delay aging or say slow it down and prolong lifespan. So we think that now the problem with removing senescent cells with race car macrophages is that not every type of senescent cell has a particular cell surface signal on it that we could target. So in order to use it against senescent cells, we probably have to make race car ends against individual types of senescent cells one at a time, rather than just universally all senescent cells. But there's pretty good evidence that getting rid of senescent cells can be beneficial. One of our best friends is CEO of a company that's working on a drug that targets senescent cells in macular degeneration, and it looks promising in like phase 2 trials. So I think senescent cells would be one way that we can use that, and then eating up and eliminating things like tau tangles and amyloid plaques, and using it, prophylactically, I think is possible.
Any more questions? I'm sure that you're considering lobby or somehow communicate with people in DC to express the importance of the work so that they will continue funding it. That's like a job all by itself.
That's true. And so I think that, the question was, like, are people trying to communicate with folks in DC to lobby for continued support of this kind of research? And I think in this moment, some of our scientific societies can be really important because they are bigger than any one of us and bigger than any one institution. So I was a president of the Genetic Society of America in 2020 and sort of got a good look behind the scenes at like how societies function. And so there's the American Association for the Advancement of Science, the American Society for Cell Biology, the Genetic Society. There's a whole bunch of societies, and they are practiced, like they have infrastructure and people in place already who lobby on a regular basis. So I think they're really well positioned to undertake that. We in this room are in a really great position to undertake the effort to lobby our California legislators. And just today, there was an announcement that one of our state senators is, what's his name?
Scott
Weimer.
Yeah.
Scott Weimer, is proposing a California Institutes of Health to much of the way that we have a California Institute for Regenerative Medicine, which when George W. Bush was president and limited federal funding for stem cell research, California stepped up and the populace voted to create a California Institute for Regenerative Medicine, which is really important. There's a student in my lab who is funded off of a CERN program, and it's been a really important program for California. It sets a beautiful precedent for how you can respond to a negative change in at the federal level, and we can respond at the state level. We're a big, powerful, wealthy state, and we ought to be able to become somewhat self-sufficient. And so I would encourage all of you to, if you care about this kind of research and life-saving research, to support and maybe even lobby your state legislators about that, because that's an initiative that really where we could take matters into our own hands to some extent.
Anything else? Anybody else?
Just following up on that last point, there was an article in Politico this morning that announced that Scott Weimer, the state senator, was proposing legislation for support in California to kind of replace the anticipated shortfalls in funding from the NIH. So what you can actually do something, and you can actually do a lot, is not only write if you live in this area, Assemblyman Park, and also our state senator, Moni, but even more importantly, you can spread the word, write your friends and colleagues who are in different districts to then lobby their assemblymen and assembly member, I should say, and state senators to support Scott Weimer's bill. So again, if you just Google his name, Scott Weimer, W-E-I-M-E-R. And this topic, you'll find articles in Politico. And so I think there's something you can do. It's actionable.
Great. Well, thanks for your attention. Yeah. Oh, great. You're going to be a macrophage. Yes.
That was me.
Okay.
I've missed, I'm a retired teacher, and
Yeah.
I'm gonna start crying. I've missed the kind of.
Yeah.
That was wonderful.
Great.
You did a
Thank
great job.
you. Thank you.
Denise Wilcox.
Yes, 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 of course. Yes.
Say good job.